All right, guys, check, check, check. Biological psychology. We are on the visual system. The visual system. Seeing versus perceiving. So, seeing versus perceiving. I never really knew this, but seeing is sort of in the family of sensory. Seeing, sensory, right? I didn't really think about that. So, the visual system, seeing versus perceiving. Sensory system, the eye. Exactly what we sense from our environment. The eye, what we sense from our environment. Perceptual system, to perceive, right? Seeing versus perceiving. And so we have the perceptual system, which is the brain. It helps us to perceive the brain. How we put together what we sense into a visual picture. So how we put together what we sense. So the eye is exactly what we sense. And when it comes, that's the sensory system, which is the eye. Exactly what we sense from our environment, and perception, and the perceptual system, the brain, how we put together what we sense into a visual picture, has to do with perception, perceiving, how we put together what we sense into a visual picture. That's done through the brain, but our sensory system goes through the eyeball. So let's continue on with the visual system. Balance syndrome. It is a parietal issue. That is the part of the parietal lobe of the brain, which is the somatosensory system. It typically has to do with touch. So that balance syndrome results from parietal lesions, dorsal, wear. Certain symptoms from the balance syndrome are the ocular apraxia, difficulty shifting gaze, fixating, following movement. Gaze tends to wander. Ocular apraxia, gaze tends to wander. Optic ataxia, inability to reach for items under visual guidance. So ocular apraxia, ocular, so difficulty shifting gaze, fixating, following movement. Somebody's gaze tends to wander. This is balance syndrome. Optic, think about optics, inability to reach for items under visual guidance. Simultagnosia, unable to focus on more than one thing at a time. Unable to focus on more than one thing at a time visually. That's balance syndrome, has to do with it's a parietal issue. Wear results from parietal lesions, dorsal, wear. Symptoms, ocular apraxia, difficulty shifting gaze, fixating, following movement, gaze tends to wander. Optic ataxia, inability to reach for items under visual guidance. Try to remember optics, inability to reach for items. Ocular, difficulty shifting gaze, fixating, following movement. The gaze, somebody's gaze tends to wander. Simultagnosia, unable to focus on more than one thing. Think about simultaneous. You could do both of those things at the same time. So unable to focus on more than one thing at a time. And you got optic. You got inability to reach for items under visual guidance, and the ocular, difficulty shifting gaze, etc. Then we have prosopagnosia, inability to identify familiar faces. So these are like these are problems when you have problems with the visual system. Prosopagnosia, inability to identify familiar faces. Can identify the person by voice. Can identify gender, age, and emotions of faces. So can identify gender, age, and emotions of faces. Led to the FFA debate. So prosopagnosia, the inability to identify familiar faces, that led to the FFA debate, which is expert versus face area. Scientists argue that the FFA is not sensitive to faces, but instead is sensitive to identification of things that we can expertly identify. We've all seen so many faces that we are effectively face experts. Bird watchers show increased activation when identifying various types of birds. There was something that I wasn't understanding when it was being taught in class, and maybe on Thursday I could get a sort of a better breakdown from the professor. And now there's something called greebles. Train people about the different types and check their activation. Major problem, they are face-like. Greebles. Train people about the different types and check their activation. Major problem, they are face-like. I gotta ask him about that too. Greebles and the FFA debate. Greebles and the FFA debate. I have to come back during the next video and maybe clarify that for you guys. So how does the eye work? A key part of the eye is the retina. You can't see without the retina. So the retina changes light into electrical signal. So the retina changes light into electrical signal. Translates light waves into an electrical signal our brain can process. I guess it's a concave, curved. Object on retina is translated upside down. Within the retina, there are photoreceptors, which are rods and cones. There's an optic disc, also known as the optic nerve. It's an area in the retina where nerves and blood vessels exit the eye. It forms a blind spot. And there's something called the fovea. It's an area in the center of the retina. The fovea is an area in the center of the retina. Optic disc, optic nerve, area in the retina where nerves and blood vessels exit the eyeball, forms a blind spot. The optic nerve, optic disc, is an area in the retina where nerves and blood vessels exit the eye, forms a blind spot. And the fovea is an area in the center of the retina. So it's gonna speak about rods and cones right now, but let me take a guess. Rods are the black and white, and the cones have to do with color. So layers of the retina. There are four main types of visual processing neurons. Four main types of visual processing neurons. There's the ganglion cell layer. There's the amacrine and bipolar cell layer. They're horizontal cells. Visual sensory neurons have photoreceptors, which are made up of the rods and cones. So rods and cones, where a sensory signal, light waves, gets changed into electrical energy. So transduction. Where a sensory signal, which are also known as light waves, get changed into electrical signals that our brain can process that. It's a process called transduction. So rods are sensitive to black and white. Most of them are in the periphery of the eyeball, or the side of the eyeball. And cones are sensitive to color. Most of them are in the fovea, the center of the retina. The direct pathway. So you go from the photoreceptors, which are rods and cones. That's where that's how our vision processes light, chemical signals that come through the brain, or they come through the vision. So it starts out with the rods and cones, then hits the bipolar cells, then reaches the ganglion cells. The bipolar and ganglion cells contain receptive fields. They are antagonistic center-surround organization. there's somebody called Stephen Kuffler's work. He recorded from ganglion cells in cats, and these always fired at some baseline rate, and they could be excited or inhibited. The ganglion cells always fired at some baseline rate, and they could be excited or inhibited. So I guess Stephen Kuffler shined circles of light in various places of these cats' eyes, I guess, and he found that receptive fields were smaller in center of the eye and larger in the periphery. The receptive fields, when he shined circles of light in various places, he found that receptive fields were smaller in the center, it says or the eye, in the center of the eye, and larger in the periphery. So the center of the eye would be the retina, fovea, periphery. Something else going on there. Periphery, I think, is the retina, right? No. The retina is actually the retina is like pretty much, no, it's not just the retina. The center is the fovea. Fovea is the center of the retina. And the periphery, we say there's more in the periphery. There are more rods. So the periphery is more black and white, and there's more cones in the center. In the fovea, more cones. So color, black and white. So center, surround. So it's sort of kind of drew a picture. It was kind of hard to understand. If you're looking at the center of something, there's more circles in the middle. Like you're more focused at the center of your vision. But on the outside, they're more bigger donuts. So it's more of a blur. It's not as focused, right? If I'm focusing on something, everything else around it is not as focused, right? It's not as precise. So he drew a picture right here. It's kind of at the center and then surround. And you got the receptive fields. And we have this here. I know it's hard to see because it's kind of during the day where you can see. So let me study this. So diffuse illumination, no change. Change in activity when you focus. So on center field, excitatory center, inhibitory surround. Spot on center, light on. Spot on surround, less activity. Center illuminated, happy. Surround illuminated, unhappy. Diffuse illumination, no change in activity. So off center field, inhibitory center, excitatory surround. So also inhibitory, so excitatory surround. What's an excitatory surround though? I don't really understand. So we got excitatory on the outside, surround illuminated. Surround illuminated. We don't like it because if it's excited in the center, but everything else outside of it is illuminated. I don't really want to show you guys, but So if on center field, right? On center means the center is excitatory. So if that's the spot is on center, it's just a spot, specific spot. So you're focused in on it. There's activity. But if it's spot on center, but the surround is excitatory, there's more activity on the outside. Spot on surround. So ganglion cells, on center bipolar fields connect to on center ganglion cells. Off center bipolar fields connect to off center ganglion cells. So off center, on center, on center. Obviously, bipolar cells or bipolar fields connect to ganglion cells, but if it's off center, bipolar fields they connect to on center. On center bipolar fields connect to on center ganglion cells, and off center bipolar fields connect to off center ganglion cells. Other parts of the retina. Other parts of the retina. Horizontal cells and amacrine cells integrate information, play a role in inhibition of the bipolar and ganglion cells. So the horizontal cells and amacrine cells, they integrate information and they play a role in inhibition of the bipolar and ganglion cells. What do receptive fields do? On-center and off-center fields provide for greater acuity. Large, unchanged surfaces don't activate the neurons, as well as changing surfaces with lines, cracks, and ends. Edge detection. Big to small. Convergence of information. 120 million rods and cones. Only 800,000 axons in each optic nerve. Fovea has the least convergence. Close 1-to-1, center. Periphery has the most, which are the rods, black and white. Convergence also occurs as bipolar cells connect to ganglion cells. More convergence in periphery and less in fovea, less in the cones. Color. More convergence in the periphery, which are the rods. Visual pathway to brain. Optic nerve leaves the eye through blind spot. Some information goes to opposite side. Optic chiasm. 80% of axons go to thalamus, go to the LGN, the lateral geniculate nucleus, which gets sent to primary visual cortex V1, the occipital lobe. The remaining 20% of axons go to brainstem structures involved in sleep, eye reflexes, and eye movements before going to the thalamus. Contralateral organization. Opposite side. Visual hemifields in space are contralaterally organized. Not the contralaterally organized by the eyes. Eyeballs not contralateral. Visual hemifield, where you're looking. I feel like something I should know there. So if I look at something, right? So if we see something, your brain, if I see something on the right, that information gets sent to the left side of my brain. So that's the contralaterally organized. The eyes don't do that. The eyes The eyeballs actually process it on both sides. But the brain processes it differently. Visual cortex. Hubble and Weissel in the 50s and 60s tried to find receptive fields in the primary visual cortex of the cat. Started with light spots like Kuffler had done previously, got no response from these cells. On accident, passed a line over the receptive field and got the cell to be active. They passed a line over the receptive field and got the cell to be active. Still organized in centers around organization, just not circular anymore. Still organized in centers around organization, just not circular anymore. Receptive fields in V1, the primary visual cortex, is organized into columns. V1 is organized into columns. Remember that these cells like lines. Layer 4 of V1, simple cortical cells, respond best to lines in a particular location with a particular orientation. So orientation is how it's oriented diagonally and where it falls within, let's just say like a square, if it's on the right side line. So layer 4 of V1, simple cortical cells, respond best to lines in a particular location with a particular orientation. V1 is organized into columns. Layers 2, 3, and 5 in the same column, complex cortical cells, respond to a line in a particular orientation over a group of locations. Orientation columns, because all of the cells in a column respond to the same orientation of line best, they are called orientation columns. Because all of the cells in a column respond to the same orientation of line best, they're called orientation cells. What happens when you look at the rows of cortex? Each 0.05 millimeters of sideways movement through the cortex leads to a 10-degree rotation shift for the preferred line orientation. Approximately 1 millimeter of cortex is needed to get all line orientations 180 degrees worth. All line orientations within this unit would be from the same location, just the orientation of the line is different. So again, perception is processing, seeing is sensation, the eyeball, and perception is the brain. So visual processing after V1, over 30 different cortical visual processing areas. Each seems to add something to the picture. V5 is visual motion. V4, color processing. So V5 is visual motion. Anyway, there seems to be over 30 different cortical visual processing areas. Each seems to add something to the picture. V5, visual motion. V4, color processing. Now, a couple other people. Unger, Leiter, and Mishkin, 1982, reviewed lesion studies with monkeys. Temporal equals sound, auditory. So if the temporal lobes were lesioned, the monkeys cannot learn a pattern. If the temporal lobes were lesioned, the monkeys cannot learn a pattern. Discrimination task, but could still snatch flies out of the air. If the parietal lobes were lesioned, they can recognize objects, but cannot accurately reach for them. So if the temporal lobes were lesioned, what? So if the temporal lobes were lesioned, cannot learn a pattern, discrimination task, but could still snatch flies out of the air. If the parietal lobes were lesioned, can recognize objects, cannot accurately reach for them. Two pathways emerged. The top, the dorsal pathway. Where pathway. The bottom is ventral, the what pathway. So the temporal lobe has to do with vision as well with auditory. So two pathways emerge. The dorsal is the where pathway, and the ventral, the what pathway. So where to look, or where something might be, and what. Let's talk about some sort of issues, some visual problems. So visual agnosia, it's a brain perception issue. Patients have normal sight according to an optician. Two types, two different pathways of visual agnosia. Apperceptive, basic vision is intact, cannot combine the elements into something. Vision but no perception. So visual agnosia. There's apperceptive, it's having your vision intact but cannot combine the elements into something meaningful. So they could see but they cannot perceive it. So there's a damage to where regions, damage to the where regions. They can see but cannot combine elements into something. They cannot perceive. Associative, cannot copy an image fine. Or associative, can copy an image fine. Vision but no perception, cannot identify it or extract information about it. Damage to the temporal regions, the what regions. As where is the apperceptive, the associative is the what regions. So parietal, where, temporal, the what. All right, we have a vision activity handout. We'll go over this real fast. So for the table below, what type of stimulus is being processed at the location given? So again, we're going to start from the beginning. Is where that sort of electrical signal that was changed from a chemical sort of sensory information comes into the eyeball and gets processed in the brain. So cells in the eyeball, the cells we have are rods and cones. We have the bipolar and ganglion cells. So rods and cones have to do with so rods process the black and white information. The cones process the color information. The bipolar and ganglion cells have to do with those spots and circles, on center, off center. And you have simple cells in the V1. Simple cells have to do with line orientation plus location. So let's say there's a square, where the orientation of the line and where it's located. If you're seeing it directly in the center of that sort of object, or like pretty much focus. So simple cells in the primary visual cortex V1 area has to do with line orientation and location. Complex cells in V1 have to do with only line orientation. A bunch of lines within that. Yeah, so they respond to a line in a particular orientation over a group of locations. The dorsal stream, where objects are, so sort of how to navigate your world visually. Don't run into things. So the dorsal stream has to do with where objects are, dorsal, and the ventral stream has to do with what something is, what is it being perceived. So the dorsal stream is the what, or no, the dorsal stream is the where. Remember where objects are and how to navigate your surroundings. The ventral stream is what is being perceived, what something is. So how does the term convergence apply to the visual system? So remember, you start off with 120 rods and cone cells. That's when that electrical signal is being sent through the eyeball structure, comes through the rods and cones, black and white's being processed, colors being processed. The periphery has to do with the rods, black and white. The middle is the fovea, the retina. The middle of the retina is the fovea, and it has a lot more cones. It processes color, and that's more of a one-to-one processing system. So those 120 million rods and cone cells get broken down to fewer bipolar cells, and from there it gets broken down to even fewer ganglion cells. There's less than 1 million ganglion cells, and then it gets pretty much sent out. So it's hard to see this, but you have an eyeball here, right? This is the front of the eye. You're looking out this way, and then from the sides, you have rods on the sides of the eyeball. Well, this is actually the top. We're looking at the top of the eyeball. We have an axial view of the eyeball. So we're looking at the top. So these right here, these are rods here. These are rods. So the top of the eye, or the sides of the eyeball are the rods. So that's black and white information. And then the middle of the eyeball is the in the middle of the retina is the fovea, and that has to do with remember, color. And so millions of rods go into fewer ganglion cells. There's a lot more convergence with the black and white, because the black and white's mostly not, doesn't have to be as detailed as when you've seen actual color, right? The actual color has to do with the cones and the fovea. So that has less divergence, because you don't need a lot of more blur or a lot more. It needs to be precise, so it's more of a one-to-one convergence. So cones are one-to-one to ganglion cells. Rods are many to fewer, so rods have more convergence. That's the idea of convergence. Is the visual system contralaterally organized? Yes, it is, but it's based on visual hemifields. So let's say we stared. Look at my hands right here, right? But look at me, right? You're looking directly at me, but what you see on this side, it's getting processed in the left hemisphere, in the brain, right? You're looking at me though. So right here is getting processed on this side of the brain, right? And what's over here is getting processed on this side of the brain. You're still looking at me though. Look at me. And so rods and cones are more over here, because it's, I don't know, the more the rods, the black and white blurriness is more over here, right? And so, but your left eyeball can see a little bit. You're looking at me, but your left eyeball can see a little bit of the right and the left, right? It could see, so that's processing. It could see both sides. So it's not just contralateral. It could see both sides. It's a little hard to understand, but as long as I know right here to get the answer right on the test. And so it's not like one side can only see one side. It processes both. And so what it sees on one side, it's processing it on the opposite side. So if I cover my eye, my right eye can see a little bit over here. So it still processes what over here on this side, and it's processing what's over here on this side. So the eyeball is not just contralaterally. You could see both sides, but the brain is processing it on, I don't know, it's sort of difficult to understand. If the answer is like, is the maybe I could figure out problems and be like, is the visual system contralaterally organized? Yes, but not the eyeball, right? The perception might be contralateral, but not the sensory. The eyeball is not contralateral. Anyway, so the fovea and cones. The fovea and cones. So the color are the cones. So the color, which is, oh yeah, no, the fovea is the cones. Remember, it's the rods is what see color. They're more on the periphery. But the fovea, which is the center of the retina, has more cones, has to process color, more detail. And because it needs a lot of detail perception, center of the eyes, information coming from center, you want it to get through. So that's why it's more of a one-to-one convergence. And the rods, which are the sides of the eye, which are the periphery, not as much detail. So there's a lot going on in the sides of the eye. It's okay to lose some of the detail perception. Information condensed in the ganglion cells. Well, the ganglion cells are also processing cones as well, but it's more of a one-to-one convergence from the cones to the ganglion cells, but there's a lot more divergence from the peripheral rods diverging into the ganglion cells. Anyway, so that's the end of the visual system video.